Yeah, good afternoon once again, and I welcome you all for the fourth session of this Indo-Korea joint webinar on advances in biosensors. And this last session, the talk will be delivered by Professor Jung Heon Lee from SKKU, South Korea. And the title of the talk is Synthesis and Functionalization of Nanomaterials for Biosensing Applications. And I'm very happy to introduce Professor Lee. Dr. Jung Heon Lee is an associate professor at Sungakon University, Korea. He got his BS in metallurgical engineering from Yonsei University, and then MS in material science and engineering from Seoul National University, and then PhD in material science and engineering from the University of Illinois at Urbana-Champaign. After postdoctoral training at Northwestern University, he joined the School of Advanced Material Science and Engineering at the SKKU as a faculty in 2012. He is also affiliated with Biomedical Institute for Convergence at the SKKU, the Institute of Quantum Biophysics, and the Research Center for Advanced Material Technology at SKKU. Professor Lee is leading the Biological and Nanoscale Materials Laboratory at the SKKU, where he is focusing on the development of nanoscale materials or devices which can be applied to diverse levels of biological systems for diagnostics as well as therapeutics. He is also investigating opportunities to adapt programmable and readily modifiable properties of biomolecules in the fabrication of smart materials or advanced nanoscale devices. He has published more than 85 research articles in the peer-reviewed international journals and filed 50 patents, including 7 US patents and 5 international patents. With this brief introduction, I request Professor Lee to deliver his talk. Professor Lee, over to you.
Okay. So first, I appreciate Professor John to give me a chance to co-organize this webinar between India and Korea on advances in biosensors. Okay, so let me go through. So let me first introduce about SKKU. So this is what the SKKU actually looked like a long time ago. So this school was founded in 1398 as a Royal Confucian Academy. And the first library was established in 1475, and it became a modern university in 1895. And in 1953, it became a comprehensive university. So this is one of the old palaces, Changdeokgung, which is located in Seoul. And old SKKU is located just behind this Changdeokgung Palace. And so the SKKU Seoul campus is located just next to this old SKKU. So it's just on the backyard of Changdeokgung Palace. So about the historic legacy, so in Korea we have 4 bills, and all the people, so we have 4 peoples on each of the bills, and all 4 peoples are related to SKKU. So we have this very famous, so King Sejong. He's the one who literally invented the Korean alphabet by himself. But he was the chairman of board of trustees of SKKU. There was this person, Yi Hwang. So he's a Confucian scholar, but he also used to be statesman and politician. but he served as president of SKKU. This person is, he's called EE. He's also a Confucian scholar and statesman and philosopher. And he used to attend SKKU as a student. Then there is this lady, so she's called Shin Saimdang. But at that time, women could not attend college, so university. But this lady was a mother of this EE. So she was the parent of the student who attended SKKU. So anyway, but now SKKU is so owned by Samsung Corporation, and SKKU and Samsung is having close relationship between each other. It's similar to Sastra University, having connection with like Tata Corporation as well. That's what I have heard about. So we have two campuses. One is located in Seoul, and in Seoul there are colleges so on the humanities and social sciences. While in Suwon campus, we have colleges related to the natural sciences, including School of Medicine and School of Pharmacy. So this is the ranking of the program of material science, and so SKKU is ranked around 29th based on this QS World World University rankings, and it is comparable to very renowned like universities, including University of California, Santa Barbara, University of Michigan, and here there is Caltech as well. So we are really working very hard to be comparable to these universities. Okay, so let me introduce a little bit about the connection we had between Professor John at Sastra University and myself. So we first could be able to be connected through Dr. Shankar, who was like Professor John's student. So he got his PhD under the guidance of Professor John, but he so came to our lab as a postdoctoral scholar. And then we could be able to have the spark project between each two. So this is the picture we took when Professor John came to SKKU on November 2019. And then after that, so I myself had a chance to go to Sastra University. So this is myself with like two students. And then I had a chance to teach in Sastra University as well. So these are the students who attended the class. And after that, we had an MOU between Sastra University and the SKKU. So the College of Engineering at SKKU. And then, so Professor John and I had a chance to so publish a book together on the biomarkers and biosensors for cervical cancer diagnosis. And this book is now available at Amazon, and the price is $180. And this is actually comparable to one of the most expensive books that I have ever bought before. Okay, so just okay, so let me now move on to the main topic. So in our lab, so we are in material science. So we are mostly kind of focused on developing materials which can be used for biological systems. But we are also like so interested in using adapting biological systems, so molecules, to material science as well. So here, so I just introduced like different research areas in our group. So one part is to develop nanomaterials which can be used for biomedical diagnosis and treatment. This one is on materials which can be used for tissue engineering. So we are working on the ECM mimicking materials and also bone mimicking materials, etc. as well. The third part is on bio-inspired materials. So in this case, we are trying to adapt biomolecules or biological systems to material side, but I will mention about this a little bit later. And also we are also interested in using artificial intelligence to design materials and also to characterize them as well. So here is one thing which falls into the third category of the bio-inspired material. So Professor Uma just mentioned about ATP as the molecule which produces energy in our body. But in our lab, we try to adapt this ATP as a flame retardant material. So this is a bare polyurethane, which is one of the most widespreadly used thermal insulation material. But as you know, they're really flammable material. So if we just, you see, they just burn very easily. But if we simply dip coat this material using ATP, oops. Yeah, so simply this polyurethane foam doesn't burn. Okay, so we believe this flame retardant property of ATP comes from its unique property containing 3 phosphate groups, 1 ribose sugar, which contains carbon, and also adenine, which contains a lot of nitrogen. So what this phosphate group does is once it becomes burned, it will produce phosphate groups and metaphosphate, which will interact with this ribose sugar and form char. This is the char. So this is ATP pellet, and once we burn it, you can see like char. So huge char formed, and this char will be able to coat the polyurethane surface, and it will protect the polyurethane from being burned. Okay. And also it contains this adenine group, which contains a lot of nitrogen. So what happens is once it is burned, ammonia will be formed from this adenine group. And so this will be able to remove the oxygen from the polyurethane. So it will kind of do the fuel dilution effect. Okay, so this is one of the work we did on the flame retardant material. But actually, once Professor John comes next month to SKKU for his sabbatical, actually we're going to work on the development of electromagnetic shielding and flame retardant advanced textile materials. So this can be one of the work which is connected to that project. Okay, so let me then move on to the main topic today. So in our group, we are interested in working on the optical biosensors, and gold is one of the material which is used very frequently on those applications. I think everyone likes gold, and I mean, this was like this from a long time ago, from ancient time as well. They look very expensive, yellow, but once they become very small, so they have different colors from red, pink, so purple and blue, and this depends on their size. And actually, these gold nanoparticles interact with light in different ways. So they can either absorb light or reflect light. So we call that observance and scattering. And whether the nanoparticles absorb light or scatter light depends on their size. So if the size of the nanoparticle is small, they dominantly absorb light, while if the size of the nanoparticles becomes larger, they dominantly scatter light. So why is the gold nanoparticle red? So this is the extinction spectra of gold nanoparticle. But once so white light is shed to the nanoparticles, what happens is that so this is the extinction spectra of the gold nanoparticle. So this color will be absorbed by gold nanoparticle. So we will be able to see the remaining colors, and you can see among the remaining colors, so this red color is dominant. So that's the reason gold nanoparticle looks red in many cases. However, this extinction spectra is different, like for different size of nanoparticles, and that is the reason these nanoparticles look in different colors at different sizes. But these nanoparticles do not always look red. So once they scatter, so if white light so interacts with nanoparticle, again we have extinction spectra here. So once the nanoparticle scatters the light, we will be able to see the light which is reflected from the nanoparticles. So the color which is reflected from the nanoparticles are, in this case, so mostly green color. So that is the reason the nanoparticles, so spherical nanoparticles, gold nanoparticles looks green. So if the nanoparticles with the size of 50 nanometer scattered, they look green, while if the nanoparticles, if the light is transmitted from the nanoparticles, they look so pinkish. This so color of the scattered light is different depending on their size, component, and shape, etc. For example, gold nanoparticles with spherical gold nanoparticles with the size of 100 nanometer look like orange, while again the 50 nanometer gold nanoparticle look so like green. So spherical silver nanoparticle with the size of 100 nanometer looks white, so which is different from this. So as silver nanoparticles become smaller, they have different size as well. And if we compare these two, like with similar size but with different shape, you can see that their color is different as well. So this is like a very famous Microscope, which is from the Romans. And you see, even though they are like identical cup, they have different colors. So in the case of the left side, so this this Microscope looks like this when the light source is from the outside. So if the light is scattered from the cup, you will see a green color. While on the right side, if the light source is inside the cup, what we will see is the color transmitted from the nanoparticle. So in this case, we will see red color. Okay, so and also depending on the shape, so if the nanoparticles are spherical, again we will see red color. But as the nanoparticle becomes longer, so in the case of nanorods, they will produce green color and also brown color as well. So you can see here that as the shape of nanoparticle changes, their color becomes different as well. So that is the reason, like a lot of research groups are developing different materials with different compositions, different size, and different shapes, because they all have different characteristics. Also, aggregation of nanoparticle is an important factor because once they become aggregated, the plasmonic coupling of the nanoparticle happens. So if nanoparticles are well dispersed, again, they have red color. But if they become aggregated, so they will, so size of the nanoparticle will become larger. So the scattering will become dominant, and the plasmonic resonance will shift to the red side. So if we chemically functionalize the nanoparticle with biomolecules such as DNA, then it's possible for us to produce nanoparticles functionalized with DNA molecules. And then if we have two batches of nanoparticles functionalized with two different DNA molecules, they will remain red because there is no specific interaction. So assuming we have no specific interaction between the DNA molecules. But if we introduce a third DNA, which is complementary to DNA 1 and DNA 2, this third DNA will induce hybridization of the DNA molecule, which will eventually aggregate the nanoparticle. So through the interaction between the DNA molecules, we can induce the coupling of the nanoparticles, and we will eventually see their color change. I previously worked on a system where we can take advantage of this to do metallic ion sensing. So in this case, we used this DNA zyme, which is specific for uranium oxide ion. So in the absence of these molecules, what happens is these nanoparticles are aggregated. So they will produce bluish purple color. But in the presence of uranium oxide, this one will induce the cleavage of the complementary sequence, which will eventually help to release the nanoparticle. So through this, we will be able to see the color change happening from blue to red in the presence of this uranium oxide. You can see here that in the absence of uranium oxide, the nanoparticle is purple, but in the presence of uranium oxide, it is colored red. And in between, we can see the color change happening from purple to red. This is a wonderful system in a sense that we don't need any a complicated instrument to do the detection. So we can just do it simply with naked eye. But the main issue of the colorimetry sensor is that they have relatively poor sensitivity compared to other systems. And also, another issue is that it's very difficult to distinguish the presence of uranium oxide if if very small amount of uranium oxide exists in the sensor. So this is one of the issues that we wanted to solve in the next part of this talk. What we wanted to do is to develop a sensor which works in yes/no type colorimetric sensor. So this is the way normal colorimetric sensor works. So color changes from one chromatic color to another chromatic color, and in between these two, there are a lot of colorimetric, a lot of colors as well. So we focused on this color wheel, which is based on CMYK model. So, and we focused on the fact that if we mix two colors which are complementary, then it's possible for us to produce a black color. So we thought that if we add a dye which is complementary to this color, we will be able to produce a black color, which is achromatic. But eventually, the color of this sensor will change to red because what we added is this orange color, and then this greenish color was supposed to change to this color in the presence of certain analyte. So we thought that through this strategy, we will be able to develop a sensor whose color changes from achromatic color to chromatic color. So we can develop a yes/no type sensor, which allows us to do intuitive analysis using naked eye. So we first checked whether this might work using Benedict solution, which can detect glucose, which is something that we discussed in the previous Professor Uma's talk. So Benedict solution itself works like this. So in the presence of glucose, you can see the color change happening in between these regions from 10 millimolar to 30 millimolar. However, the detection limit of this sensor is 2 millimolar if we measure it using UV-vis spectrophotometer. So you can see here that the limit of detection from our naked eye is much lower than that of the limit of detection which we get from instruments. However, if we simply just add a complementary dye, in this case, it's this color, then eventually we will be able to produce black. In the present, if we simply add glucose, its color will change brownish. And then if we do this, you can see that color change occurs in between these regions from 1 millimolar to 3 millimolar, which is actually 10 times higher than this case without the complementary color dye. You can see the color change happens in between these regions, and this is actually comparable to the limit of detection we got from the UV-vis spectrophotometer. So this shows the possibility that we can increase the sensitivity of this sensor close to the level of the UV-vis. And also this allows us to do the intuitive detection. So we can simply, very easily recognize the presence of the analyte using our naked eye. So after that, we looked into the possibility to adapt this method to nanoparticle-based sensor. So in order to do this, we used three different nanoparticles having different colors. So red colored gold nanoparticles, yellow colored silver nanoparticle, and cyan colored gold nanorod. So if we just simply mix them, we thought that they will produce black color. And then if we add target molecules 1, 2, 3, which can interact with DNA 1, DNA 2, and DNA 3, and cause aggregation of each of the nanoparticles, then we hope that it might be possible to induce color change from black to green, purple, and orange. We try to do this, and eventually we could be able to prove that we can eventually do that. So the color change happens from black to light green, light purple, and light orange in the presence of different target molecules. So this tells it is possible for us to do multiplex DNA detection. So in the first case, you can see that the color changes from black to orange, and color change starts to happen around 0.5 nanomolar, which is comparable to the limit of detection we got from UV-based spectral photometer. While in the case of case with only 1 nanoparticle, so it's very difficult to see the color change. So this again shows that it is possible for us to do this on-off type detection using naked eye. So with enhanced sensitivity. What is also like noteworthy here is that because we in this case brought like another parameter. So normally what people do is they use RGB as a measure to quantify colors, but that's actually quite complicated. But in this case, we start from achromatic color. So if we measure the saturation as a new parameter, it's possible for us to do much sensitive detection, and that's what we could be able to do using our system. So we can, if we just simply take an image using our smartphone, it is possible for us to do quantitative measurements through this in a much sensitive manner. We could also do detection of biomarkers such as thrombin or PDGF. So it is possible to do multiplex protein detection using this method, and this also works in human plasma as well. And so I mentioned about the functionalization of nanoparticles using DNA, but it's also possible to do that using inorganic material as well. So this is a study we did by functionalizing the surface of the gold nanorod with mesoporous silica material. In many cases, very difficult to control this. But by like in this case, we really tried very hard to set up the condition for the nucleation and growth process. And through this, we could be able to produce very monodisperse, nanoparticles with core shell structure with very small amount of these core-free nanoparticles. You can find one here. So this can be used for like drug delivery applications, for example. This is another example where we have, in this case, again, mesoporous silicon nanoparticles, but inside we put like many nanoparticles inside. So here we have gold, so like several nanoparticles inside the mesoporous silicon nanoparticle, but it's possible for us to change the density. So here they are all focused inside, but in this case they are well dispersed outside. And it is also possible for us to place gold nanorods, quantum dots, and iron oxide nanoparticles all together. So this is the TM image of them. So by controlling the density of these nanoparticles, if they are focused inside, you can see that so the plasmonic coupling of the nanoparticle can happen, and also the fluorescence of the quantum dot will be quenched. So if they are all focused inside, but if they are well dispersed, this plasmonic coupling doesn't happen. but the fluorescence signal of the quantum dot can be enhanced by the gold nanowells. So it's possible for us to control the optical properties of these nanoparticles as well. So this, we believe, can be used as a multifunctional therapeutic agent as well, which can be used for drug delivery, but also we can do the diagnosis at the same time. Okay, so let me move on to the next part on the electron microscopy-based single DNA analysis, which was done in collaboration with Professor Kyubong-jo, who talked in the second talk today. I thought that he won't talk about this. He briefly mentioned about it, but I will talk about the whole story so you can have a better idea on it. Like imaging, so like molecules such as like protein. So recently, there was a study on using TM, prior EM to analyze the protein got Nobel Prize. But like imaging something is really important. In our case, we were interested in like imaging and also analyzing genetic DNA molecules. So here we have 46 chromosomes, and DNA is actually very long. So if we stretch all the DNA, so they are around 2.2, they become as long as 2.2 meters. In the case of the first chromosome, which is the longest among them, so their size is around 8.5 centimeters. However, DNA molecule itself is around, diameter of a DNA molecule itself is around 2 nanometers. So in order to analyze DNA, we need an analytical instrument which not only has a very high resolution, but also has the capability to allow us to kind of image large areas. So TM has been used before to analyze DNA. So here I brought several studies which were used to analyze, observe DNA molecule using TM. But DNA is good in a sense that it's very like elegant instrument. So we can do very precise detection, but it's very difficult to like image large area using this TM. While in order to however, there is like this fluorescence like microscope is used like often to analyze DNA molecule. The good thing of this fluorescence imaging is that it's possible to do imaging on large scale. But the problem of this is that the resolution of this fluorescence is like a couple of hundreds nanometer, which is much larger than that of the resolution of the DNA. So we thought that scanning electron microscope, SCM, can be one of the instruments which we can use to overcome these problems. So here I show you some examples of like the studies which used SCM to analyze image the biological like systems. But in the case of SCM, in order to use SCM, since we are imaging through the secondary electron emission, since the secondary electron emission is needed to observe using SCM, we have to coat these samples using the nanoparticles, using metals. So in all of these cases, the metals are coated. But in order to visualize DNA, so we have to basically coat the DNA with metal. So that's the difficult part because DNA itself is very small. So here I brought several studies which report metallization of DNA using metals. But you see in many cases, in most cases, they are not continuous. So it's not ideal to use them for the analysis of DNA. In most cases, what they do to do the metallization is they first treat the DNA with metal precursors, and then they reduce these metals using reducing agent. So what they do is they do the nucleation and growth process in situ. We thought that when they do this, the problem is they won't be able to produce enough nuclei, which is necessary to grow these, to do this metallization. So what we did is we used this DNA binding peptide, which Professor Zhou mentioned in the previous talk. So this one contains 1, 2, 3, 4, so lysine and 2 tryptophan. So these lysine can interact with DNA backbone through the electrostatic interaction, while this tryptophan can intercalate into the DNA bases. And then if we place a thiol group on one end of the DNA, we thought that it would be possible for us to introduce a lot of these thiol groups on along the DNA molecule. So basically, if we have DNA molecule, if we treat this DNA binding peptide containing thiol group onto it, so this can be work as a binding site to attach gold nanoparticle Cs on it. And then if we further do reducing process, we thought that it will be possible for us to grow nanowire along it. So this is the image of the nanoparticles which are attached on the DNA. So you can see that they are well aligned along this DNA, while this doesn't happen in the presence of non-binding peptide, which is a control peptide. It's possible for us to image DNA using nanoparticles, so using SCM as well. And then if we do the metallization, we could see this monomer, dimer, trimer of the nanoparticle of the DNA. So we thought that this kind of works well. And also it's possible for us to kind of image DNA which are entangled. And this is actually very difficult to do using other analysis techniques such as TEM, AFM, or using fluorescence microscope. Okay, then let me move on to the next part. So we thought that we just showed an example where we can do DNA analysis. So we can image DNA by doing metallization using DNA binding peptide. But we thought that this can be used as a new method to attach functional groups along materials such as carbon nanotube. So Professor Uma just talked about using carbon nanotube and graphene as electrode for the electrochemical detection. But normally, it's very difficult to introduce functional groups on the surface of the carbon nanotube. So what people do is they normally treat it with strong acids such as sulfuric acid. But that will induce kind of unwanted defects on the carbon nanotube, which will work in negative way. So what we did is we first treated carbon nanotube with single-stranded DNA, and since this single-stranded DNA contains a lot of bases which has ring structures, it will be able to attach on the surface of the bind to the surface of the carbon nanotubes through the pi-pi interaction. And then after that, if we treat this carbon nanotube wrapped with single-stranded DNA with DNA binding peptide, this will be able to, so the DNA binding peptide, since, so again, this DNA binding peptide contains lysine and tryptophan. Lysine will be able to bind to the DNA through the electrostatic interaction. The tryptophan will be able to bind not only to DNA bases, but also on carbon nanotubes through the pi-pi interaction. And also, it can bind to the, so the binding can also happen to the through the cation and pi interaction as well. So once they bind, if we introduce thiol group, carboxyl group, and biotin on the surface of the on one end of the DNA binding peptide, it will be possible for us to attach gold nanoparticles, magnetic nanoparticle, and quantum dot to the carbon nanotube surface specifically. So we have shown that this actually works very well. So we can kind of evenly attach gold nanoparticles, magnetic nanoparticles, so carbon nanotubes, quantum dots on the surface of the carbon nanotubes. And we kind of demonstrated the application of this nanoparticles for catalase activity, for decomposition of the hydrogen dioxide. And so we have shown that this the decomposition level is the highest when the nanocomposite containing all three nanoparticles works all together. It's much higher than the cases with 1, 2, or 3, 1 or 2 nanoparticles. And we have shown that this happens through the synergistic catalase effect, which happens from in the presence of gold nanoparticles, so quantum dots, iron oxide nanoparticles, and carbon nanotube. So let me wrap my talk. So first, I want to, so this is my group, and then we have here Dr. Prof Shankar, who I mentioned previously, who kind of connected the two groups in Sastra University and SKKU. So Mr. Mutu in Professor John's group visited our group kind of 2 years ago. And then now we have so Shruti and Priyan in our group. They are kind of doing their doctorate so studies in our group, and then they actually just started. So collaborators, so there were a lot of so very good collaborators, but I want to acknowledge Professor John for this wonderful collaboration, and also Professor Zhou, who worked on the second project that I mentioned today, and then to Professor Kwang Il Lim, who will be talking tomorrow. So with Professor Lim, we actually worked on a project to detect so avian flu virus using fluorescence method, but I didn't talk about this at this time. And then I appreciate the funding, so which allowed us to do the our research. So National Research Foundation of Korea. Then these two, all of the funding agency, and also especially so Spark project, which allowed us to kind of do this collaborative so program. Okay, thank you very much for your. Oops, I brought a video here. So we kind of brought a drone, so you can have an idea what SKKU looks like. So this is the library, And then the engineering building, which where I'm looking, so I am working at, is located here. Okay, so thank you very much for your attention.
Professor Lee, for your excellent talk on the functionalization of nanomaterials for biosensing applications. I have one question. You have linked the DNA with the gold nanoparticle and worked on S or no type, like chromatic to achromatic and vice versa. Have you tested whether the change in color with reference to a specific DNA, which is, say, a cancerous one, which is, say, for example, a cervical cancer and a normal cell? Is there any significant difference you have observed?
Oh, that's one of the projects that Shruti has been working on. And then indeed, she could be able to see the color change. So using that method. Yes, so indeed, yeah.
Okay, yeah, that would be really interesting when we have with a normal cell and a cancerous cell or a DNA with.
Yeah.
Okay. And I'm just checking, is there any? So any questions from the audience, please post your questions in the chat box. Okay. Okay, then, yeah, again, once again, Professor Lee, we thank you so much for your nice talk.
Thank you very much.
And again, I thank all the delegates, students, scholars who joined us for the whole day for the 4 sessions. And again, I invite you all for the tomorrow sessions. There will be 3 sessions tomorrow. The first session in the morning at 9:30 by Dr. Pranjal Chandra from Indian Institute of Technology, BHU. Then my second session tomorrow at 11 a.m. by Professor Kwang-il Lim from Sukmyong Women's University, Republic Korea. And of course, the last session will be myself at 2 o'clock. So I invite you all to join for the 3 sessions tomorrow. So once again, thanking Professor Lee. Let us close the today's activities of this 2-day Indo-Korea joint webinar on advances in biosensors. Thank you, Professor Lee.
Thank you very much.
Thank you.
See you tomorrow.
See you tomorrow. 1 second, Professor Lee.